A paper came out last month that caused quite a stir in the longevity community. In a study out of Israel, they significantly extended mouse lifespans through the activation of SIRT6. Now, there's already been a bunch of videos on this paper, but I wanted to think about it for a while before I put out my own video and integrate it with data from other studies that I've read. Well, I've done that, so let's dive in. Okay, here's what's coming up. First, we'll start off with a quick recap of what sirtuins are, what they do, and how they function. Next, we'll talk about the new paper that came out last month on SIRT6 and how it extended lifespans in mice. Then we'll be looking at activators of SIRT6 and Fucoidan in particular. And we'll wrap it up with some news about where you can obtain a form of Fucoidan that will actually activate SIRT6 and potentially extend your own lifespan. Lately, there's been a lot of buzz around SIRT6, which is one of the sirtuins. Just in the last few months, there's been papers published about SIRT6, senescence, and age-related cardiovascular disease, the role of SIRT6 in cancer, SIRT6 and its neuropathic function in age-related loss of cognition, SIRT6 as a master regulator of immune function, the positive effect of SIRT6 on multiple hallmarks of aging in the hearts of mice. And this paper screens several activators and inhibitors of SIRT6 in the treatment of breast cancer. And then there was a paper published last month, in May of this year, that claimed to have significantly extended mouse lifespans through the activation of SIRT6. And this paper has been creating a lot of buzz. So is Dr. Brad Stanfield and the Shiki Science Show. Articles have been published on lifespan.io and on Longevity Technology, as well as a slew of other websites dedicated to longevity. So actually, I'm kind of late coming to the show, but the paper had major implications, and I wanted to think about it for a while before I put up my own video on it. And honestly, it's taken me a couple of weeks to get my thoughts in order about how I wanted to integrate this study with information that I've obtained from other studies. And I want to start out by reiterating that I'm not a doctor or a scientist or a researcher, and that anything I say should be taken with a grain of salt. It's entirely possible that I may be mistaken in my interpretation of this study. And if any of you guys have a different take on it, be sure to let me know in the comments. But before we get into all of that, I think we need to do a quick review of what sirtuins are and how they function. Sirtuins are silent information regulators. And once a second sirtuin was discovered, the word sirtuin was coined by combining the acronym for silent information regulator with the number 2, SIR2N. In humans, there are 7 sirtuins and they all belong to the same enzymatic domain. Actually, sirtuin is the name given to both the gene located in our DNA and the enzyme that it expresses. These 7 sirtuins are labeled SIRT1 through SIRT7. They're all structurally related, but they perform different functions. And because of those structural differences, they react with different proteins. Sirtuins 1, 6, and 7 are located in the nucleus of the cell. Sirtuins 3, 4, and 5 are found in the mitochondria, and SIRT2 is located in the cell's cytoplasm. Now, all 7 are NAD+ dependent, meaning that their enzymatic function depends on the availability of NAD+, or nicotinamide adenine dinucleotide. Sirtuins in general are responsible for maintaining homeostasis within the cell, particularly energy homeostasis. They control inflammation, they boost the efficiency of ATP production, but one of their primary functions is the repair of damaged DNA. Now, as I understand this, the way they do this is actually pretty simple. If NAD levels are high enough, and this is important, so let me repeat it, when there's enough NAD+ available, sirtuins can act in a couple of different ways. Either in the removal of acetyl groups from proteins, primarily histones, and this is called deacetylation, or by adding one or more molecules of ADP ribose to proteins, called ADP ribosylation. Now, this alters the location, the activity, and the stability of these proteins. So some of the functions that sirtuins perform are done through the deacetylation process, and some of them are done through the ADP ribosylation process. And it's the ones that are done through the ribosylation process that are of particular importance here, because it's through ribosylation that SIRT6 recruits PARP1, which is also NAD plus dependent, to facilitate the repair of damaged DNA. Now, while all the sirtuins are involved in the homeostasis of an individual, a couple of them seem to be more relevant to human health. One of those is SIRT1. So scientists have developed a certain kind of mouse called a transgenic mouse that can overexpress various sirtuins. Overexpressing SIRT1 has been shown to improve the health span, but it doesn't seem to have much effect on the overall lifespan. Now, there is one exception to this. Overexpressing SIRT1 in the brain of mice has resulted in mice that can live longer. So SIRT6 is starting to look like the sirtuin that is the most active in slowing down the aging process and promoting longevity. Animals that have longer lives have higher levels of SIRT6. For example, while creating transgenic mice that express extra SIRT1 didn't really have any impact on longevity, overexpressing SIRT6 in mice has resulted in longer living mice. SIRT6 has a beneficial impact on several of the hallmarks of aging, including genomic instability, telomere attrition, and epigenetic alterations. Boosting SIRT6 also reduces inflammation. SIRT6 silences something called line 1 transposons, which are genetic parasites that are one of the main causes of inflammation. It's also neuroprotective, and people with Alzheimer's have low levels of SIRT6. SIRT6 also increases the efficiency of ATP production, which is vital to energy management. As you age, you lose metabolic homeostasis. And this happens in both mice and in humans, and both show changes in energy expenditure and metabolic flexibility. Now, think about it. A lot of the interventions to promote longevity do so by activating mitochondrial function and energy metabolism, because it's in the mitochondria that the energy that the body uses is created in the form of ATP. Now, this suggests that enhancing energy production might be beneficial in extending the human lifespan. And one of the essential metabolic processes for creating energy is gluconeogenesis, or GNG. GNG creates glucose, which is one of the body's primary sources of energy, from non-carbohydrate precursors. Now, this happens primarily in the liver. There's a lot of data that demonstrates SIRT6's role in metabolism. SIRT6 impacts gluconeogenesis during fasting, which is one of the times that GNG is really activated. SIRT6 restores age-related deterioration in GNG capacity. It promotes catabolic and anti-inflammatory pathways in the liver. SIRT6 maintains NAD plus levels, and it increases the synthesis of NAD plus. In a study done back in 2012, overactivation of SIRT6 extended lifespans in male mice, but not in female mice. It turned out that these mice had lower levels of insulin-like growth factor 1, or IGF-1. Now, this is important, and I'll come back to it in just a minute. All right, so a new study came out just a little over a month ago titled Restoration of Energy Homeostasis by SIRT6 Extends Healthy Lifespan. It was published by Professor Chaim Cohen, who's the director of the Segol Healthy Human Longevity Center at Bar-Ilan University in Israel, and he was assisted by an international team of researchers. This paper was looking at both SIRT1 and SIRT6. They created transgenic mice that overexpressed SIRT1, overexpressed SIRT6, or overexpressed both. And they also had a control group of wild-type mice. Now, this next part is a little confusing. I've watched a bunch of videos and read a lot of articles that state that they obtained a 30% increase in the lifespans in both male and female mice. And this includes a newsletter put out by Bar-Ilan University, where the study was done. But I read the paper, and it stated that overexpression of SIRT6 led to a 27% extension in the median lifespans in males and a 15% extension in females. Now, they could have been talking about the maximum lifespan that they achieved, but the paper stated that SIRT6 expression induced 11% and a 15% extension in maximal lifespans in males and females respectively. Now, that just doesn't make any sense to me. If the maximal extension in males was only 11%, how could the median extension be 27%? So I suspect a typo or something in the copy that I obtained, which came from Nature Communications. If any of you guys have an explanation for all of this, let me know in the comments. It went on to say that these mice exhibited significant improvement in overcoming a variety of age-related diseases, such as cancer and blood disorders. Mice were able to conduct the same level of vigorous activity as young mice, and they didn't become frail. Now, this study showed that overexpression of SIRT6 leads to a reduction in frailty and life extension in both female and male mice. Professor Cohen stated that aging is associated with overall decline in health and increased frailty, and is a major risk factor for multiple chronic diseases. Frailty syndrome, characterized by weakness, fatigue, and low physical activity, affects more than 30% of the elderly population. Through a variety of biochemical methods and metabolic analysis, the researchers were able to decode the mechanism through which SIRT6 extends lifespans and health spans. They found that as animals grow older, they lose the ability to generate energy in the absence of external energy sources. Now, we're talking about food here, and an example of this would be during a short period of fasting or calorie restriction. So what we're talking about here is that reduction in gluconeogenesis that I was describing earlier. Wild mice have an age-related decline in both glucose homeostasis and in liver glucose output. Old SIRT6 transgenic mice were able to preserve both glucose output and glucose homeostasis by improving utilization of lactate and glycerol. To do this, SIRT6 increases gluconeogenic gene expression in the liver, NAD+ synthesis, and it systematically enhances glycerol release from adipose tissue. These transgenic mice that can overexpress SIRT6 can maintain an enhanced energy production capacity from other storage, such as the breakdown of fats. This creates glucose that can be utilized for energy in the muscles and specifically in the brain. SIRT6 activates a physical response that is identical to diets that promote longevity. Diets include fasting and calorie restriction. So we're talking about energy, right? And we get our energy from the nutrients that we consume. This study also showed that inhibition of the major nutrient-sensing pathways mTOR and insulin IGF-1 could extend lifespans in organisms from yeast to mammals. At the same time, activating pathways which can sense a low energy state, like the AMPK pathway and sirtuins, could also lead to increased longevity in a wide variety of organisms. And of all the sirtuins, SIRT6 seems to be leading the pack here. Well, that's all well and good for transgenic mice that have been created to overexpress SIRT6. But what about us? We don't overexpress SIRT6, and I don't see any path in which we will. Well, it turns out that activating SIRT6 will produce similar effects to overexpressing it. So we need to find some compounds that will activate SIRT6. So let's talk for a minute about inhibitors and activators. Inhibitors work better and are more common than activators because they only have to block the substrate receptor sites. Now, an example of a substrate would be NAD+. But there's a type of sirtuin activation that's called allosteric activation. This is when a compound activates a sirtuin by binding to a different site than the sites that a normal substrate would bind to. Researchers have found a number of compounds that have been reported as sirtuin activators, and these compounds are called STACs, or sirtuin activating compounds, and these are allosteric activators. Now, some allosteric activators turn on the deacetylation process, and some turn on the ribosylation process, and some turn on both. And most STACs are compounds that we've talked a lot about before: resveratrol, pterostilbene, fisetin, and quercetin. But most STACs are better at activating SIRT1 than they are at activating SIRT6. Now, I have read that quercetin and fisetin will activate the deacetylation function of SIRT6. So will a compound called MDL800. But finding a compound that will activate the ADP ribosylation process of SIRT6 has proved to be much more difficult. And finding an activator of the ADP ribosylation process of SIRT6 is important. That's the process through which SIRT6 is able to repair damaged DNA through the recruitment of PARP1. It's also through the ribosylation process that SIRT6 is able to reduce levels of IGF-1. In fact, as I understand it, it seems that most of the life-extending properties of SIRT6 are through the ADP ribosylation process, not the deacetylation process. That's where Professor Vera Gorbanova comes in. She's a co-director of the Rochester Aging Research Center at the Rochester University Medical Center in Rochester, New York. Her info page on the university website lists over 100 papers that she's either written or co-authored. Now, she and her team have been investigating SIRT6 and SIRT6 activators, and she's claiming that a new activator of SIRT6 has been found in Fucoidan, which is extracted from a type of seaweed called Fucus fasciculosus, commonly known as bladderwrack, rockweed, sea oak, and cutweed. Now, interestingly enough, Fucus fasciculosus was discovered in 1811, and it was the original source for iodine. So, fucoidan is a long-chain polysaccharide, the most common form of carbohydrates found in plants. It's most commonly found in brown algae, like Fucus fasciculosus. But it's not as simple as just eating a bunch of seaweed, even though that's what people from a number of Asian countries have been doing for centuries, and they're noted for their longevity. But as it turns out, not all fucoidan is created equally. While some types of fucoidan activate SIRT6, most of it actually blocks SIRT6 from being activated, and there's no way to tell which is which. So they've been collecting samples from all over the world and batch testing each sample to find out which samples activate SIRT6 and which samples block activation. Now, when they finally find a sample that activates SIRT6, they then extract the fucoidan from that batch. But they often find that all the samples that they collect block activation. When that happens, they have to toss out all the samples and start over from scratch. It's a lengthy and labor-intensive process.